All right, hey y'all, let's talk about the cell cycle and mitosis. So, we just finished up learning about DNA and really just a basic knowledge of what is the structure and its base pairs. Now, we know DNA is found in the nucleus, and something that takes place in all of our cells to sustain life is the cell cycle. It's so important for you and I. It is how our bodies heal ourselves, it's how we grow. It is really what allows us to sustain life in general. When cells are not doing what they should be doing, it's because this cycle is off in some way. So this is where we can go into areas where we learn about cancer and things like that. But we need to have a basic understanding of the cell cycle, not for your Cambridge curriculum, but for the end of the year state test, because the cell cycle is something that's thoroughly covered. So let's dive into it. The cell cycle is the series of events that cells go through as they grow and divide. So this is how cells ultimately divide to recreate themselves, to produce new cells. All right. Now we can look at the different phases of this. So the cell cycle, and let me scoot this over. The cell cycle has several phases. We say it starts with the G1 phase, and this is all a portion of interphase. Interphase is the period of growth that occurs between cell division, and ultimately it includes the G1, the S, and the G2 phases. So all of this side with the G1, S, and G2 phases, that is referred to as interphase. Now, when we look at specifically what are the G1 phase, the S phase, and the G2 phases, what's going on in your cells during the G1 phase of interphase? This is a phase in which the cells do most of their growing, and cells will increase in size and then synthesize new proteins and new organelles. So it's duplicating everything. Then we can move on into the S phase. The S phase is where we have chromosome replication and DNA synthesis. So once that DNA replication is complete, the cell then enters into the G2 phase. During the G2 phase, many of the organelles and molecules required for cell division are produced, and then when the G2 phase is complete, we say that the cell enters the M phase, or the mitosis phase. Mitosis is the portion of the cell cycle where really all the intense stuff is going to go on. You can see some really cool things under a microscope. So let's look at mitosis specifically. I have pulling out a new graphic behind the screen here. So cool. Okay, so let's look at mitosis. We can divide mitosis into 4 phases followed by cytokinesis. And these phases are prophase, metaphase, anaphase, telophase. If you see the letters that start with those, you might hear the little mnemonic device PMAT. So the phases of mitosis: PMAT, prophase, metaphase, anaphase, and telophase. And this is what's going on. So here, interphase with the G1, the S phase, and the G2 phase. Everything has been prepping for this big divide. So one cell can become 2, okay? Now we enter the mitosis phase. We start with prophase. So the chromosomes appear in a condensed form, and the nuclear envelope starts to go away. So the nucleus starts to kind of disappear in a way. That nuclear envelope starts to disappear, and you can now see where your chromatin of DNA was all jumbled up and all loosey-goosey in the center here. Now you can start to see defined chromosomes. They have condensed themselves into those little X shapes that we think of when we see a chromosome in a science book. Then we move on to metaphase. Think meta, down the middle, metaphase. So the thick coiled chromosomes, each with their 2 chromatids, so one side of that X, chromatid, we call them sister chromatids, they're so cute, okay? They line up down the middle, okay, and you can see they're right in the middle. So metaphase, think middle. All right. Then we move on to anaphase. This is where the chromatids of each chromosome begin to separate and pull apart. What's actually happening, these little guys right here on the corners are called centrioles or centromeres. They are going to attach to the center of your chromatid, to the centromere of the chromosome with something called spindle fibers. So those reach out, attach to the middle of those cute little sister chromatids, and start pulling them apart. So anaphase, apart. We're moving away from one another. Those sister chromatids are separating. So see you later. Adios. And then we move on to telophase. Telophase is where the chromosomes are at opposite poles or opposite ends and are becoming more diffuse. The nuclear envelope starts to reform. So you can see this nucleus is starting to reform itself around the DNA material that's replicated and now in its own individual sides. All right. And then we start to see that the cytoplasm starts to divide. And then we don't get that full division until we have cytokinesis. We can call this the nuclear cleft or even the nuclear cleavage. So once they actually divide, that is the process of cytokinesis. The cytoplasm divides and boom, we started with one cell here up at the top and now we have two. And that is cellular division. All right. I'll catch y'all after y'all take y'all's notes for the next CFU on cell division.